Hello, everyone, and welcome to today's presentation titled Unlock a New Era of Automated Mini and Maxi-Scale Plasmid Purification with AmMag Quatro Solutions. This webinar is part of the 9th annual event in the Laboratory Automation Virtual Event Series. I'm Matt Thorne of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, go to genscript.com. Before we begin, I'd like to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located on the left of your screen. If you are having any trouble seeing or hearing the presentation, you can click on the Help Desk button located at the top of your screen within the navigational bar or from the lobby within the event. Now let's go ahead and get started. I now present today's speaker, Luciana Rosselli-Murai, Head of Field Application Scientist Team at GenScript. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Luciana, you can now begin your presentation.
Thank you very much for the introduction. So I'm Luciana. I'm the head of the Field Application Scientist team here at GenScript, and I'm very happy to deliver this webinar for you today. So very briefly about my background, I did my PhD in molecular and structural biology. And then I joined University of Michigan, where I passed many years doing research over there as a postdoc, and I was studying signaling pathways in cancer cells. So now, not now, but many years ago, I joined GenScript, and what I'm going to present today is part of the work that we do here at GenScript. So the two instruments that we're going to present today is to automate your mini preps and your maxi preps. So to begin my presentation, I'm just going to go through the contents of the presentation. I'm going to give you a brief introduction, background. I am going to talk about specifically about the two GenScript solutions, the AmMag Quatro Maxi and the AmMag Quatro Mini. And then we're going to talk about the application data, and we're going to have the Q&A after that. So to start, just to tell you a little bit about GenScript, it's just a quick slide here, just to show that we were founded in New Jersey in 2002, and since then we have grown quite a lot with many other companies being under a GenScript umbrella. So about the plasmid itself. So let's talk a little bit about the history of the plasmid. It's just very interesting to know that in 1952, the term plasmid was first introduced. But very late in the 80s, several new techniques was developed to extract the plasmid and purify plasmid DNA. These methodologies actually would take days to accomplish, and it's very interesting that to know that, because these days we can purify a plasmid DNA in 2 hours. So So the purification methodology itself, that is just an overview about the purification. So you can start with the lysate preparation. So there are many methods to lysate the cells, and the method the most commonly used is the alkaline lysis. What are the benefits of lysing alkaline lysis? So it's very effective to remove protein, genomic DNA, and RNA from your prep. It's very easy and the definitive methodology to be used. In our automation, it uses the alkaline lysis as well. So after you have your lysate, you are going to proceed with the purification of your plasmid DNA. So there are many methods to purify your plasmid DNA from the lysate. So the methods out there are ethanol precipitation, that's still very commonly used, cesium chloride ultracentrifugation. This is a methodology when it takes a long time to perform, but you want a high quality of plasmid DNA and 100% supercoil. There are also the boom method, the silica membrane kit, and the most used one, that is the anion exchange columns. That is most of the current manual solutions use the anion exchange columns. However, our method is the magnetic silica beads, and I'm going to explain in the next few slides how we implement these in the automated machine. So here is the workflow, the current workflow if you are doing traditional purification of plasmid DNA. This workflow, many of you might know very well, that involves several steps, includes the resuspension of a pellet, the lysis, neutralization, centrifugations, binding, washes, and final dilution. All these steps require a lot of centrifugations and passing the samples through the columns, and the columns can clog, and you cannot handle so many samples at the same time. So thinking about this workflow for plasmid purification, GenScript has worked for many years to automate this whole process. And the way we did that was only possible because we have the know-how of making beads. So the beads for plasmid purification has a ferrite core that is coated with silica. So using cheotropic agents, the plasmid DNA can bind to these beads. So the advantage of using silica beads and beads itself for purification, it is actually very advantageous because you can skip the centrifugation steps. Why? Because the beads, they can achieve the solid phase reversible immobilization that we call the CPRI by just applying a magnet field next to the sample, and the magnets are going to be immobilized and separated from the solution. So this feature make it possible to implement automation through all the steps of plasmid purification that would include in the binding, the washes, and the elution. So here I'm showing you in this slide the two solutions that GenScript developed for you. On the upper right side, you can see the AmMag Quatro Maxi. We called 1400, and for short, we can just call it Quatro Maxi. So of course, by the name, you can know that this instrument is for Maxi preps. So the other instrument that you launched is very recently is the Quatro Mini. So the Quatro Mini instrument is for automating your mini preps. This is on the bottom on the right side of the slides. So these two instruments are fully automated and we're going to talk about details about these two instruments in the next slides. So I'm going to first start with the Quatro Maxi, and I'm gonna, before I start talking about this instrument, I'm gonna just show you a quick commercial video so you can have a better idea how it works. So the video you just saw was made a long time ago. So the version of the controller was the older version of the controller. Now we have a very small controller, as you can see on the right side of the slide. So the controller for the Maxi-Prep instrument can be connected to 4 modules, 4 Maxi modules. So these, each one of the modules can run up to 6 samples. So if you have all 4, you can run at once 24 samples. And GenScript also provide a complete solution because we also provide the kits and the consumables as a kit for performing your Maxi preps. And all the programs are pre-verified. So next, I'm going to show you some features of the Maxi Prep instrument. So the Quatro Max, you can purify up to 24 samples if you have all 4 modules. Here is a view of inside of the instrument, and you can see some of the features that you also saw in the video. So in the very front, you have the waste bin, and then you have the first Quatro collection tubes. That is where you're going to put your pellets. So after you grow your culture, you centrifuge your culture inside of the tube that we provide in the kit. So this first tube here with the cap on is a 50 ml tube, special designed for the instrument. So we're going to centrifuge our culture inside of this tube, pour out the media, and then we are going to put this tube insert inside of the instrument. The next lane of tubes, we call the reaction tubes. Before we start the run, we are going to fill this tube with 100 absolute ethanol or 200 proof, 40 mLs of it, and then inserting inside of the instrument in the second row of tubes. And then after that, we are going to have to insert the reagent cartridge. So the reagent cards come with all the buffers you need for one prep, meaning that comes with the resuspension buffer, the lysis buffer, the neutralization buffer, the pre-clear beads, the binding beads, the binding buffer, the binding beads, and all the endotoxin removal buffers and wash buffers, and also the final elution buffer. Everything is contained in one cartridge. And this cartridge is very easy and ready to go. And you just need to slide into the rack and place the rack on the back of the instrument. And finally, you have your tip boxes that come with 6 tips, all with filters, and the final collection tube. All these has to be added to the instrument, loaded into the instrument on the very back. And your final purified plasmid DNA. After you start your run, it's just a few clicks, you start your run, and after 2 hours and 20 minutes approximately, you're going to have your purified plasmid DNA transferred to the final collection tube, this one with the green lid. So after 2 hours, you open the instrument and take that tube from the instrument, centrifuge for 5 minutes, and then you have your purified plasmid DNA ready to use. So talking about the features of the instrument, the pipetting accuracy of the instrument is 97%. So this is the coefficient of variance of 3%. And in terms of the culture volume that you can process, it just varies because depends on the media that you are using. If you are using a very rich media such as TB or Plasmid Plus, that is a commercial available media, probably you can use about 60 or even 25 of Plasmid Plus media is enough to get the optimum pellet weight that you can process through Quatro. But if you are using media that's not so rich, such as LB, so then you might need to use 200 mLs of LB to get to the same pellet weight that is optimal for Quatro. So the runtime is 2 hours and 20 minutes approximately. This is for the most used protocols. I think the longest protocol we have so far is 2 hours and 20 minutes. And what we're going to get is a transaction grade plasmid DNA with the endotoxin levels below 0.1 EU per microgram, supercoil ratio above 80 to 90%. So, and also, as you can see on the bottom of the slide here, you have all the consumables ready to use, and we provided this as a kit, and you don't have to provide anything else. So we have everything for your prep ready to go. So let's move on. This is the workflow for the Maxi instrument. Everything that is in this rectangle here is everything that is fully automated. The only two parts, as I mentioned, that is not automated is the centrifugation of your cultures and the last step of spinning when you have your plasmid DNA in the insert above the inside of the final collection tube. So you just need to spin it down. So your plasmid DNA is going to go through endotoxin removal resin. That is the last step of removing endotoxin from your sample, and through a 0.22 micron filter. So your sample is going to also be sterile filtered. So, and this only takes about 5 minutes to complete, and you have your plasmid DNA ready to go. Just going through the workflow of automation here. The first tube where you put your pellet, the pipette arm is going to transfer the resuspension buffer, the lysis buffer, and the neutralization buffer. And then after that, the clear beads, we call clear beads, but because these are the beads that bind to all cellular debris, proteins, genomic DNA, this is going to be incubated with your sample. So you will see by the end of this step that all that precipitated proteins, that white stuff, is bound to the beads. So after that, the magnet comes in place. The beads are attracted to the magnet, and the clarified lysate now is transferred to the next tube. The clarified lysate is then incubated with the binding beads. The binding beads are the beads that bind to your plasmid DNA specifically. And then you have, after the binding, there is an incubation time, and everything in all the steps is modifiable in the software. So that is the beauty of the system as well. So you can modify the parameters to get higher yield. You can get also more concentrated or less concentrated plasmid DNA as you wish. So the next step is just the wash steps, and always the instrument perform always in the same way. The magnet comes in place, the beads are attracted to the magnet, and then all the wash buffers are going to the waste. The final step is the elution, but before the elution, you have one step that we call the air dry. So that step is to the tube rotates clockwise and counterclockwise for some minutes that you can set up in the protocol. That's to remove the excess of ethanol from your sample. So then your elution buffer is dispensed there, and then your plasmid DNA is go into the solution, and then the magnet comes again, and the eluate is transferred to the next tube, the final tube. So you have your purified plasmid DNA ready to be centrifuged and ready to use. Okay, this is just the workflow of the whole process. And here is just a couple of screens of the interface, the software interface. It's very easy to use. Here on the left, upper left, you can see the first interface. That's if you have all modules connected, you would just see all in blue, dark blue. In this case, you only have D1, device 1 connected. So would you click there to get access to the system. You can go to the settings, you can edit your protocols, you can export the data, you can go to the general sets or upgrade your software. You can also, the bottom left image screen is the screen where you scan all the components before you start the run. Just take about 20 seconds to happen, and it's very easy to use and very nice to have this function as well. So that after that, you can just click and start your protocol, choose your protocol and it starts your run. So after that, you can check in the status, and you can see this kind of workflow, and is highlighting and is showing which step you are in the run, and also going to inform you the remaining time. So next, let's talk a little bit about application data. So this first application data is one side-by-side comparison that we did very early on, that where we compared the Quatro with the manual prep. In this case, I was using the QIAGEN kit. So we used PCDNA 3.1 and PCDNA 3.4, they're high-cap plasmids, and we did the elution with a Quatro with 600 microliters of volume, and then 500 for the QIAGEN prep. In terms of concentration, what we got was a very similar, a little bit lower than with this protocol compared to the QIAGEN kit, like about 1,000 nanograms per microliter. And the 2.6/280, that is telling you about protein contamination, was in the normal range for both, for all the samples. And the 2.6/230, or the reagent contamination, was also in the normal range. Then the toxin levels was all below 0.1 EU per microgram. And the supercoil ratio, as you can see in the gel above here, was all above 90% for all the samples. So moving next to the next application data, we decided to test two culture medias, LB and TB. So for this study, we used the same volume of TB and LB, that was 100 mLs of either or, and then we processed through the same protocol. So we looked in both samples in the same. That was done in triplicate, of course, and we eluted with the same volume. And here's the concentration that you got, meaning that if you have more plasmid DNA because you have more pellet to start with, so you're going to have more plasmid DNA. So this means that in this case, TB, you get higher yields because you have more pellets to start with. And all the other QC parameters was in the normal range, and as we would expect it to have. Moving to the next application data that we have here, that was using a very rich media. This is commercial available. We do not sell this media. That is Plasmid Plus. So Plasmid Plus, with only 30 mLs of Plasmid Plus media, we can get very high concentrated plasmids. Of course, they translate to a very concentrated yield as well. And all the parameters, the purity parameters, was in the normal range as well. Moving to the next, this is one protocol optimization that we did here in the lab. So in the instrument, you can select the default elution type, or you can select the what we call the non-separation elution. The non-separation elution, all the beads are going to be transferred to the final collection tube. This way, there is no volume loss, so you can recover almost 100% of your eluate. In this case, you can see here the results. So the same, so using the standard elution type and the optimized or the non-separation elution with this same samples, just doing in duplicate, you can see here the final volume was almost double, and the concentration was the same, so the yield was doubled by just choosing one different method of elution. So here, this is the final set of slides about application data. This was performed for one of our top customers. And what they did, they split the culture in two, and then half they process through their regular manual kit. And then the half they send to our lab here at GenScript, and it will process through Quatro. So as you can see here, the concentration was a little bit higher with the protocol that they used there for the manual kit. And our plasmid concentration was about 1,000 nanograms per microliter. All the other parameters were normal. So moving next, we send the plasmid back to them after purification. And then they analyzed by diagnostic digest, as you can see in the agarose gel here. And they did the double digest and single digestions. And you can see that the profile, they analyzed 3 plasmids in total with manual and Quatro purification. And the profile in the gel is exactly the same, and no observable RNA contents by the gel, and neither genomic DNA analyzed by agarose gels. So next, they also tested these purified plasmids on transient transfections. So transient transfection, they analyzed the titer of the protein. So here you can see that in blue is the manual kit, and green is the Quatro transfection results. As you can see, for all these 3 plasmids analyzed, the titer of the protein was exactly the same, so no difference. They also tested the same 3 plasmids purified through Quatro and through manually. They also analyzed in a platform that's a system, a platform that they do transient transfections in show. They use 96 well plate and 24 well plate formats. And as you can see by the titer of the proteins, very, very similar, so no difference on the titers. So now I'm going to move forward with the mini. So this is a new instrument that you launched very recently. So that is for automating your mini preps. So let me move through the description of the instrument. So it's also a fully automated plasmid DNA purification instrument. And that is, you're going to start from the center. You're going to grow your cultures inside of a 48 well plate. And then you are going to centrifuge this plate and insert the plate inside of the instrument, along with all the consumables that you also provide, all the reagents and the consumables for this instrument as well. You don't have to provide anything extra. Besides the ethanol. The ethanol is the only reagents that we still need the customer to provide. And then this is, there is no requirement of stopping the process to manipulate anything inside of the instrument during the run. So it's fully automated. And it's a high throughput. So the beauty of this system, in my opinion, also is that this Quatro series, you can connect to the same controller that I showed you for the Maxi instrument. You can also connect the mini instrument. So you can have one Maxi, one mini connects to one controller, or 2 Maxis, 2 minis. You can mix and match as you wish, up to 4 modules in total. So the mini instrument is for 4. If you have 4 modules connected, you can run 4 48 well plates at once. And then the run time is about 90 minutes, so it's very, very fast. And the toxin levels that you're going to get is below 1 EU per microgram. So this is the workflow that I just mentioned. You have to grow your cultures inside of these 48 deep well plates, spin it down, and then choose your protocol that you are going to run. That's also flexible, and you can change the parameters. If you want a more concentrated, less concentrated plasmid DNA, you can change that parameters. And then after that, you are gonna, after 90 minutes, you are gonna get your purified plasmid DNA dispensed in this 96 well plate, only on the first half of the plate. Okay, so moving next, this is the workflow, very similar methodology, but of course with different dimensions and for different sample sizes, because now we are processing 48 instead of 6 for Maxi preps. So starting with your centrifuged bacterial pellets plate, then the instrument is fully automated from there. It's gonna remove the media, and then it's going to proceed with the lysis and neutralization, of course, resuspension, lysis, neutralization. And you have the pre-clarification as well. The pre-cleared beads are going to be dispensed in these vessels. And then you have the magnet comes, and then you have your clarified lysate transfers to the next column in the same container. And then you have the binding beads being dispensed there. So your plasmid DNA binds to the beads. So now you have to wash those beads to reach high purity of your plasmid DNA. So you have several steps of washes that is performed in the same fashion, and all the liquid goes to the liquid waste that is inside of the instrument. And the final step is the elution. So then after so the beads are put to the walls, and then the purified plasmid DNA is transferred to the final plate, as I showed you before. So now I will show you a quick video. Well, before the quick video, sorry. This is one example about what the protocol interface and all the parameters that you can change in a run. And one of the most common parameters that you would change, that is based on the OD or the ODV of your culture, or the now we have established already many protocols, simplified protocols, that you can only use the type of media that you are using, if it is in a plate or in a tube, and then we can run a preset protocol for that. So yes, this is just an example of that, and I'm gonna just play the short video here for you. Hopefully you could have an idea how it works, how is the setup. You can see all the consumables there, and the pipette arm moving and transfer all the buffers. And it's very easy to use. The setup is like 5 minutes, and then click the protocol you wanted to run, then walk away and just come back when your purified plasmid DNA is transferred to the 96 well plate, and you have this ready to go. So let's talk really briefly about the application data for the mini prep instrument. So here was one of the first studies that we generate, that was just a screen of many plasmids, of high copy and low copy plasmids. And as you can see here, we show what is the yield for a high COP plasmid that you typically get is about 40 micrograms. Of course, you can get more with optimized protocols. Up to 60 micrograms of plasmid DNA for high COP plasmid is quite nice. And then for also low COP plasmids, you're gonna get about 10 to 20 micrograms. So in terms of purity, in terms of the ratios, all passed and is in the normal range, as you can see in the two plots on the right. So a little bit more about this optimization for Quatro Mini. So we did some optimization using different media types. And in this study was using TB, Plasmid Plus, and LB. And as you can see on the plot, we did with different volumes from 1 ml to 2.2. As you see, as you increase the culture volume for different medias, you also increase the plasmid yield. This is for one protocol using 60 or 90 microliters of binding beads. So as you can see, you reach some saturation. So it doesn't matter if you increase the pellet weight, this is not going to increase your yield. So this is a very nice study showing that if you wanted to have high yields, you probably should be using a TB media. And you're going to see in the next slides is that the purity of these samples in LB, TB, and Plasmid Plus are quite similar. So all the 26-280 values are above 1.8, and the 26/230 are both consistently above 2. So next, we also quantify endotoxin levels for LB samples and TB samples. As you can see here on the plot, the LB samples, they present really low endotoxin levels, below 0.1 EU per microgram. And TB media, higher levels, but all below 1 EU per microgram. So this is a good study to indicate that probably if you are looking for lower endotoxin levels, you should probably be using LB instead of rich media such as TB. So we also did some diagnostic digest of these plasmids, so that we grow in LB, TB, and Plasmid Plus. And as we can see, all passed the double digestion test. Also, the supercoil of all these plasmids were above 95%, with very negligible genomic DNA contamination by agarose gel analysis. So next, I wanted to provide some information about how uniform these samples are. So running 48 samples at once, as you can see, we get under the same protocol, a very consistent concentration and yield. And also endotoxin levels for each one of the samples is very consistently below 0.1 EU per microgram with these culture conditions. Next, I want just to show you some other studies that will compare with other competitors, and showing a high COP PTT5, a high COP plasmid, PET28 low COP plasmid, and a large plasmid, a back plasmid, and you compared the yield. So you can see that our yield is very comparable, if not higher, than the two competitors analyzed in this test. And then the toxin levels also are very low, and sometimes even lower than the other two competitors in the market. And the final study that I wanted to show you is that you also used, you compared GenScript mini preps purified through Quatro with the two other competitors on to analyze the efficiency of it's being transfected. So as you can see by flow cytometry and the quantification of the HFP positive cells, that these transfections, the transfection efficiency for all these plasmids analyzed through different methodologies have the same, about the same efficiency, that is about 95%. So with that, this is my final slide on application. I'm happy to take any questions, and thank you so much for being here today.
All right, thank you very much for the very informative presentation. We'll now start doing our Q&A portion of the webinar. So if you do have any questions you would like to ask, please do so now. You can submit those in the Ask a Question box at the bottom of the screen. You can just type the question and hit submit, and then that will come through on our side. So let us start with, do you offer demos on the instruments?
Yes, we do offer demos for both instruments, and we have different types of demo, depending on what customer would like to. So one very common demo that we do here at GenScript that we call the virtual demo is a type of demo where we send for the Maxi, we send the Quatro collection tubes to you. You grow your cultures, you centrifuge inside of the tubes, and then you ship the pellets back to us. When you get the pellets here, we are going to schedule a virtual demo with you, and then we process your sample in real time. After the sample is processed, we nanodrop, we discuss the results, we discuss further optimization if it's needed, and then we send the purified plasmid DNA back to you. And you can analyze, you can run your QCs, you can analyze on your downstream application to see the quality of the plasmid. So this is one type of demo that you provide. We also provide other types of demos, that is on-site demo. You have now several other programs that you can check on our webpage.
All right, thank you. Let's go with, how long does it take to purify Mini and Maxi prep?
Yes, so as I told in the presentation, for the Maxi prep is about 2 hours and 20 minutes. This is with the longest protocol, and you can purify up to, if you have only one module, up to 6 preps. If you have 4 modules, then up to 24 samples at once. And then for the Mini, the runtime takes about 90 minutes to complete, and this is for 48 samples.
All right, thank you. Let's go with, do you provide the training or tech support for the instruments?
Absolutely. So we have provided for many years already very good technical support for the instruments and services for the instrument. We provide, including, I would say that we provide the technical support. We also help the customer on site with all the protocol optimizations until they get what they want in terms of quality, purity, yield, and concentration.
All right, excellent. So it looks like we've got time for one more question. So how about how long does it set up and operate the AmMag Quatro Mini and Maxi systems?
Yes, so it is very quick because everything comes ready to go. So the cartridge for the Maxi and all the consumables. So it's about 5 to 10 minutes. A very experienced person would do this in less than 10 minutes to set up.
All right, excellent. And so thank you, Luciana. Do you have any final comments or anything you'd like to tell the audience before we wrap?
No, I think that's all I have for today.
Excellent. So thank you for your time today and your very important research. I'd like to thank the audience for joining us and the questions you sent in. I'd like to thank our sponsor, GenScript, for sponsoring today's webinar. Please visit the GenScript booth in the exhibit hall after the webinar for more information and resources. Any questions that we didn't have time for today or those that get submitted during the on-demand period will be addressed by the speaker via the contact information you provided when you registered. The webinar can be viewed on demand for a year, so until March 19th of 2026. And LabRoots will alert you via email when it's available for replay. So we encourage you to share that email with your colleagues who might have missed today's event. So until next time, thank you again for joining us, and goodbye.